Right, so I'm sure all of you know about Leonardo da Vinci, and he was interested in how things work generally. One of his major interests was how the brain worked, and this is one of his so-called papers. The idea here was that he wanted to work out how the brain was organized, and in order to do that, he injected molten wax into the ventricular system, and at the end of it, produced this beautiful image, which all of which the details of which are identical to what we know today, even with the most sophisticated MRI scans. So this was 500 years ago, and many years later, we are still trying to understand how the brain works. Or more precisely, we are trying to understand what happens to the brain when people fall ill. And today I'm going to tell you about a program that has been driven by new technologies that have become available, which seeks to put together resources that might help us understand alterations in the function of the brain when people fall ill. So the diseases that we're studying are called psychiatric syndromes, and they refer to all those illnesses which in human beings alter the way in which they behave, interact with others, and function in normal society. And it includes many common disorders that many of you may be familiar with: mood disorders, manic depressive psychosis, addiction, schizophrenia, personality disorders, and in younger children, autism spectrum syndromes. These are now recognized as a major non-communicable disease, and in the WHO Declaration of Delhi in 2012, along with diabetes and cancer, mental illness is recognized as a major non-communicable disease. And the reason for this is that approximately 2 to 3% of the population, wherever you are in the world, is at risk of developing mental illness, and approximately 10% of those people will develop franck disease. These are prolonged illnesses that affect young adults, and therefore, in addition to their medical impact, they also have major socioeconomic consequences. Now, one of the main reasons we find it challenging to treat mental illness is that its etiology and pathogenesis is poorly understood. It's been recognized for a very long time that there is a genetic basis to mental illness. They are not Mendelian in the common sense of the word, that is a single gene. Mutation in a single gene results in disease, but nevertheless the disease runs in families, and that contributes to the so-called genetic component. There are also environmental factors that act through epigenetic mechanisms, and that results ultimately in alterations in brain function that lead to the diseases shown at the bottom of the slide. So how do we why should we bother about studying psychiatric syndromes? One of the main reasons for this is that there are a number of clinical challenges that remain in understanding and treating mental illness. First of all, we are not very clear about what are the factors that result in illness, that is the risk factors, who will develop disease, and once they develop disease, what will be the progress of the disease? What will be the course of illness? Will it be short? Will it be prolonged? Will it be severe? These are very unclear at the moment. The second thing is that, unfortunately, since the 1950s, there has been no new drug to treat many of the major mental illnesses. Many of the compounds that you hear about in the press or in the journals, these are really small tweaks on compounds that were discovered pre-1950s, and there really is a major requirement to develop new ways of treating mental disorders. And so these are some medical, clinical challenges in the management of mental illness. Now, these problems could be solved if one could address some of the key basic neuroscience questions related to mental illness. First of all, it would be very helpful if one could arrive at some sort of genetic markers, genetic basis for understanding who will develop mental illness and what will its progression be. Second, it would be very beneficial to know what are the neurodevelopmental mechanisms that are altered in the brain of an individual who is developing mental illness. It would also be very helpful, and I'm sure many of you in the audience are interested in this, as to what the cellular and molecular changes in the brain are when somebody develops illness, and also what is the response of the brain to environmental factors. Now, to address all of these questions, one would like to design a study that takes into account at least these 4 main factors. First of all, it would be helpful to not just call somebody ill and say somebody has schizophrenia, but rather to have a very precise clinical definition that allows you to, if you wish, phenotype the patient much more carefully than you can do today. The second thing would be that it would be nice to have a study that captures the evolution of the changes in the brain as its structure, as its physiology and its function changes as a person goes from being healthy to being ill. Third, it would be nice to be able to capture and study the genetic changes and the environmental contributions to illness. And finally, and most importantly, it would be nice to have a way in which one can look at the cells in the brain, observe the changes that are occurring within them, and correlate that to the development of disease. Now, in order to do this, there are many ways that you could do this, and the one that we've chosen in this study is to do a so-called prospective cohort. And what you do here is to collect a group of patients and unaffected people and to follow them into the future and periodically observe them so that you can map the development of the disease. And in doing so, what we've decided to do is to use a cohort of so-called clinically dense families. And these are defined as families in which, in addition to the patient, the proband himself or herself, there are at least 2 other first-degree relatives that are directly affected by the same illness. This is an example of a clinically dense family that we're using. You can see there are 4 generations shown over here. And in the last generation, of all the individuals of all the siblings shown over there, 3 are unaffected by disease and 4 are affected by, in this case, bipolar disorder. The 4 that are affected by bipolar disorder are also differentiated into people who are responsive to major antipsychotic treatment and some that are not. And this is also known to be a genetically inherited trait. So using such families, what we're doing is to very, very carefully phenotype them. And the phenotyping is being done at several different levels. First of all, these patients are subjected to a very detailed clinical examination, including multiple levels of neuropsych testing. And the purpose of this is to classify the illness into what model organism people might call clear, distinct phenotypes, rather than a big basket within which you put different types of phenotypes. The second thing that's being done is to look at the structure of the brain in these individuals using both structural MRI as well as functional MRI, using both resting state assays as well as task-dependent assays. And finally, brain activity is also being monitored using clinical electrophysiology, in this case, mainly electroencephalograms. So using this method, we are recruiting families of individuals into the study and carrying out an initial detailed assessment of their endophenotype, as it is called. That refers to the composite phenotype of all of those things listed over there. They're then being subjected to a serial assessment, will be subjected to a serial assessment over a 20-year period, and they will be assessed every 2 years to map the development of disease. And all of this information will then go into a digital database, which with appropriate anonymization and controls will be available to all of those who want to do meta-analysis of any type on this. So the deep phenotype cohort therefore consists of the following. Over the course of the study, over the next 5 years or so, we hope to recruit 250 families, 50 each of schizophrenia, bipolar disorder, obsessive compulsive disorder, substance abuse, and dementia. And each family, we plan to study at least 4 individuals: the patient himself or the proband, a second individual, a patient affected in the family, and 2 unaffected individuals from the family, one of whom must be above the age of risk and is therefore a genuinely unaffected person, and one of whom which is below the age of risk, and therefore one can watch the development of the disease. At this point, as we are now, we have so far recruited 67 families, and 157 individuals from this group have been endophenotyped using all of the methods that I described to you. Now, all of the clinical phenotyping methods that I described will help to explain some level of the hierarchies of how the brain works. The ultimate output of the brain, which is behavior, is a consequence of several levels of integration, starting from the molecules within a cell that then wire together to give circuits, physiology, and ultimately behavior. And the clinical phenotyping study that I just described to you will address questions related to these 3 levels: circuits, physiology, and behavior. But it is unlikely to address questions about what is wrong within the cells of an individual's brain that lead to them being unwell. For example, we would like to know that if a person is mentally ill, are there changes in the function of the synapse in this person, the synapse being the fundamental unit of information transfer within the brain. We would like to know, are there any changes in the development of the synapse or in the pruning and reorganization of the synapses during development? Are there any changes in the chemistry of the synapse, neurotransmitter release, receptors, metabolic changes? And finally, are there any changes in the function of the neuron, both in terms of its electrical responses to stimulation? Now, to get around this problem, as you know, the brain is not a situation from which one can get a biopsy or of any sort. But to get around this, what we've decided to do is to use the emergence of iPS cell technology. And all of the families and the individuals that I just described to you have agreed to have a blood sample taken from them. And using that blood sample and going through a lymphoblastoid intermediate, we have generated iPS cells from all of these individuals. Each of these iPS cells is characterized and benchmarked using several criteria, 3 key ones shown over here. They should be karyotypically normal. They should express all the known markers of iPS cells. And functionally, it should be possible to differentiate them into embryonic bodies and show that they express RNA markers corresponding to all of the 3 germ lines, nestin corresponding to the ectoderm, nodal the mesoderm, and GATA for the endoderm. Okay, so these iPS cells are then also being differentiated into a stable intermediate in the development of the nervous system called the neural stem cell, and characterization of that is shown over there. And they can also will also be differentiated and shown to be functional neurons, as I have indicated over here at the bottom, by expressing markers that are characteristic of the neuronal lineage. And the hope is that by using such resources, we will then be able to have a look at the cells of patients from this collection and ask specific questions related to synaptogenesis, synaptic functions, and the physiological properties of the neurons or glia in these individuals. And I hope that many of you will take advantage of this resource and ask your own specific questions in cell biology using them. Now, of course, there must be a basis to any changes that you discover in these cells. And in general, phenotypic variations in populations, as you heard in the previous talk, arise from one of 2 things. Either they arise from genetic differences, or they arise from environmental inputs that modify gene expression. And in order to get at the first of these, which is the genetic differences, what we have done is to carry out NGS sequencing and establish the exome sequence of all of the individuals who have contributed, from whom iPS cells have been made. And because of the nature of the experiments involved, we have specifically decided to do exome sequencing rather than whole genome sequencing. I'm not going to go through the details of the genome analysis pipeline, which has been running for some time now. Suffice to say that at the end of an analysis, so this is one particular family with bipolar disorder, and what you see is a representation of the variants that have been picked up in this analysis. What's shown in blue are all the known BPAD or bipolar loci that have been discovered either by NGS sequencing or GWAS from across the world. The ones shown in green are variants detected in this family that correspond to one of these loci. And what is very important and interesting is that all the ones shown in red are new loci that have been picked up from this family by comparing between affected, unaffected, and so on. I'll be happy to go into the details of this with anybody who would like to have more information about this. So what does a resource like this look in practice if you wanted to use it? And I try to illustrate that over here. Here is an example of another family with bipolar disorder. You can see that in a single generation, there are multiple affected people and unaffected people. And you can see that as we're going along in the study, each individual is being subjected to endophenotyping, exome sequencing, iPS cells are being generated, and NSCs are being generated and functionally characterized. And these will then be available for people who would like to ask whatever question you'd like to ask about the cell biology of these neurons. As an illustrative example again, you could compare cells from the 3 individuals marked in orange who all have bipolar disorder, as well as the green one, which is a sibling which is unaffected and at the same age. And you could then compare cell biological processes between these individuals. As an alternative, you can also compare 2 individuals that are affected by bipolar disorder shown in green and red. And the only difference between the 2 of them is that one is sensitive to a major antipsychotic and the other is resistant to it. And one can therefore ask, questions about the cellular basis of how drug sensitivity arises. So in summary then, what I've told you is about a resource that uses iPS cells to generate neural cell types and ask questions in cell biology. Importantly, it's coupled with a very detailed phenotyping of the individuals from whom the cell lines were generated so that you can correlate your results back to their clinical features. And finally, all of these individuals also have genome analysis coupled to them so that you can go back and ask what the genetic basis, if any, of the phenotypes you discovered were. So all of the information about this project is available on its website, which is this one down here. And if you go to the resources section, which is outlined in yellow at the top, you can register yourself as a bona fide person and not a bot, and you'll be able to see what the resources are. You'll be able to pick what you'd like to work on, and then get in touch using that email address over there, and we'd be happy to interact with you. So this is a project that is not possible by any one individual or any one institute even. It's a collaboration between 3 institutes in Bangalore, the National Institute of Mental Health and Neurosciences, which is where all the clinical work is being done, inSTEM, and NCBS. And the project is very generously funded by the Department of Biotechnology, Pratiksha Trust, NCBS, and inSTEM. Thank you.